there are restrictions on redistribution of GTEx data files, and so it's not really possible to expose all the information and code and data underlying the browser in the previous video, but I can say a little bit about the underlying data structures. What we've done is created this thing called gtexCisStore, which uses the G ranges of bioconductor to put metadata about genomic intervals with highly functional representations of the genome. And so in this case, we have a package called gtexWBL for the whole blood tissue-specific EQTL data, and that's in fact what was shown in the browser, even though I didn't mention that. And this structure can be used to combine information on EQTL associations, in this case between a SNP on chromosome 1 and a gene VAMP3. We didn't put exactly where that is, but we're told that it's only 371,000 bases away from the SNP. So that type of metadata is available in this store. And the store is indexed in a number of ways. The genes that are involved have been enumerated, and it's possible to find the G ranges associated with certain genes very efficiently. We also have a mapping from genomic regions and components of this store. So figuring out how to take the multi-gigabyte text files from GTEC that give us all the information about the associations and this more functional computable representation that we have underlying the browser is one of the tasks of the packages called gtexLite, gqtl stats, and in this case gtexWBL, which actually manages the data which cannot be redistributed. One of the things we have to decide on is whether this is a good architecture for that type of data, or would we rather use some other distributed data store like Google Bigtable or Apache Drill for local work, or Azure Data Factory, or some other distributed data representation. This is one case where the data have been distributed across a large number of G ranges, which can be found rapidly and loaded rapidly because they're relatively small. But there are many other ways of dealing with this data distribution and query resolution, and we need to do experiments and benchmarking to figure out which ones are going to be effective for the types of analyses we want to do. The browser, of course, has very limited use. It shows us that the data are there and shows us some of the variety of types of information that are close together in chromosome space. But it's really these sorts of objects that we want to be able to build statistical models on, and that's why it's important to have something which is relatively close to the statistical analytic framework, like a G ranges.